And Lucius is the program manager for the Contagion Science program. So we will start on time as per German tradition. In fact, a minute before. So welcome everyone again to this joint meeting, which constitutes as a annual or semi-annual research meeting for the Institute, as well as the P2P Contagion Science meeting. It's a pleasure to welcome you all. Chris has already provided opening remarks, and hopefully you enjoyed the lunch that was served. Left vegetarian, right non-vegetarian. I remember that, Lana. So I want to quickly and maybe in just 30 seconds remind you all what the P2P program is, because I think it'll help set the stage. We have two speakers today, by the way, Sam Scarpino and Anton Kornick. But the P2P program is the program that the university set up, and last year, maybe a year and a half back now, awarded 5 grants. These are internally funded grants given out to 5 different projects. And the intent of it was to have the faculty members come together to collectively try and address a problem that goes beyond a single department, beyond a single person's abilities. And we were fortunate to get one of the P2Ps. And when I say we get, it's really all the faculty members who participated. Scott Acton is here representing the engineering school. So BII, School of Engineering, the college, School of Data Science, School of Medicine are all part of the Contagion Science P2P. There are 4 others. I'm sure I can't remember the names of all of them, but there is one on precision medicine, one on brain science, one on sustainability, and one more that I forget. But the intent was really for the faculty members to come together and address problems of real importance and take the university into prominence in these areas. So this is about one year that we've finished our program, and it's gone quite well. We've tried to come together a number of times, and I think one of the early successes we have had now as a part of this is being a part of a winning team that won the CDC biosurveillance or genomic surveillance program that was established. So I think it augurs well for this initiative, and we think more of these projects will be here. We'll be able to train students in this multidisciplinary area, as well as really build strong and lasting bonds between faculty members so they can work together. So I want to welcome you all again. And with that, let me introduce our first keynote speaker, Dr. Sam Scarpino. Thanks so much for coming, Sam. I know Sam for a long time. In fact, I've really seen his career grow to an amazing level in the last 10 or so years. So I've seen amazing growth in the work he's done. Sam is currently the director of AI and life sciences and a professor of public health sciences at Northeastern University, also part of the computer science department there. Additionally, he holds appointment in the network science, global resilience, and Roux institutes. I don't know whether they're separate or not. I know the Network Science Institute. Hopefully, they're all separate. Wow. Okay. I know many of his colleagues back there. In fact, many of them work with us. Folks here, Brian and Anil, Srini, they all know Alex Vespiniani and others who are our colleagues. Sam has had a really very interesting career. Not only is he a faculty, I know he was a faculty at Vermont, but he was also a postdoc at Santa Fe Institute, and he still holds a faculty position there, external faculty position, an institute that we all have a strong association with. Sam was also quite involved during the Ebola pandemic, but later on, in discussions of data and opening the data, I remember Elaine and Sam really spearheaded this huge push in opening data after the Ebola pandemic. So thank you very much, Sam, for doing that. Recently, he served as the vice president of the Pathogen surveillance group at the Rockefeller Foundation. He has since stepped down, I believe, but continues his association with them. And he's apart from all this, he's also part of and started a nonprofit organization. I think it's called Global.health. Correct me if I'm wrong, and you can tell us a little bit more about it. So with an amazing experience, I think Sam has really been able to understand the role of computation, data science, and his own background in evolutionary biology to try and address important societal questions. Today's talk is on shape of epidemics. You might have changed the title a little bit. If you have, okay. But with that, Sam, welcome. Thank you very much for joining us. Please give a big hand.
Okay. Great. Well, thank you very much, Mata, for the introduction, and all of you for having me. This is my first talk back at Northeastern since leaving Rockefeller, and I will have to go through a few of these where people invited me as a Rockefeller Foundation vice president, expecting me to show up with the checkbook, and instead I show up without the checkbook and without the Rockefeller title. So if any of you were hoping for funding, I apologize. But oh, this is going to advance on its own, isn't it? Okay. Yes, and importantly, and part of the reason I was actually very excited to be here now, in part because there are a lot of dear friends and colleagues that I've known for years, and most of you I haven't seen since the pandemic. I am now in the provost office working to start a new institute in experiential artificial intelligence, which at Northeastern means two things. One, the experiential nature of all the work we do at the university, so things like co-op programs, training of the next generation of decision makers. And then the second is applications of artificial intelligence, but focusing on things like human in the loop AI. But the reason, well, one, because I've enjoyed a lot of the conversations today about artificial intelligence and data analysis, et cetera, but more personally, because I'm at an earlier stage of the institute building journey than all of you are. And so getting an opportunity to learn about what has worked and what you might coach me on doing differently as I go through this has been really wonderful. So thank you all very much for that opportunity. I'm going to mostly talk today about what governs the shape of epidemics, and this is something that I've been interested in for quite a while. And it turns out this is something that people have been interested in basically since we started looking at epidemics. There are passages going back to sort of Roman era descriptions of the plague where they're thinking about the rise and fall of the number of cases and trying to rationalize that. The most famous or infamous model for the shape of epidemics is what we refer to as Farr's law. And if you're not familiar with this, this is the idea that epidemic curves will be symmetric. And I think one of the things that is important and that we probably don't do enough of, and I actually have another version of this talk where I try to give 4 or 5 different examples of epidemics that caught us off guard, but if we'd bothered to read the literature, we would have not been caught off guard. For example, there are antibodies in West Africa going back to the 1980s for Ebola, and so we should not have been caught off guard by that. It's been reported in plenty of studies in the literature that these places are at risk. But there's quite a bit about the misapplication of Farr's law that Farr actually warned us about in his own paper about his law. People just seem to have forgotten that part of it. So he says that epidemics appear to be generated, and of course, anytime you hear somebody use the word appear, you should immediately be nervous, but appear to be generated at intervals in unhealthy places, spread and go through a regular course of decline, but of their cause of evolution, no more is known. If the latent cause of epidemics cannot be discovered, the mode by which it operates may be investigated. The laws of its action may be determined by observation. And I actually think this is one of the more fascinating things that I've read in the last 15 years. And part of the reason why is that a lot of the arguments we still have in mathematical biology and in epidemiology is whether we have to have a physics-style understanding in order to make meaningful predictions and intervene, or whether we can do so based on statistical models off of the data. And this is Farr wrestling with those questions before we actually had settled the matter of germ theory. If you read Farr's writings, it's pretty clear Farr was on the germ side of things, even though he says unhealthy places here. But these are the same questions that we're still trying to understand now. So what has happened with this in the 100 years since is a bunch of gross misapplications of Farr's law. One of the most famous was a paper by Bregman and Langmuir, where they were trying to forecast the duration of the HIV/AIDS epidemic in the United States. So they had data through 1987. That's the filled part of the curve. They do the Farr's law thing, which is you just assume that the left-hand side and the right-hand side are going to be symmetric, so you can use a simple Gaussian, and then you can project out the rest of the epidemic. And they determined that HIV/AIDS would be gone by 1995 under no intervention. Obviously, that did not come to pass. Unfortunately, we didn't actually learn much about the meaningful failure behind Farr's law from that failure of prediction. Here's a paper published looking at Farr's law to project drug overdoses and mortality epidemic in the United States. This was published in 2014, with the red line here being a peak estimate in 2017 for when they think the peak in opioid deaths would occur. And then a steady decline, it ends here in 2035, so probably by 2040. Every year since 2017, we've had more opioid deaths than the years before, so it actually has not peaked yet. So we have another example in 2014 of the failure of this kind of reasoning. And then during the pandemic, we have this application of Farr's law to COVID-19 forecasting that was championed by the Institute for Health Metrics and Evaluation, where they did exactly the same thing that Farr talked about. They did exactly the same thing that Langmuir did with HIV/AIDS. They did exactly the same thing with opioids, which is to assume that the left-hand side and the right-hand side of the curve are going to be symmetric and generate your forecast. And interestingly enough, so one of the things that was unfortunate about IHME is they didn't come up in the tradition that a lot of us did with forecasting, the CDC challenge, et cetera, and understanding the importance of publishing your forecasts in a stable state so they can be compared, saving your data that were used to generate the forecast so that you could go back and try to retrospectively understand what happened. We learned a lot of lessons in the kind of 10 years leading up to COVID-19 and how you build good forecasting models and a good community of practice around forecasting. Fortunately, somebody on Twitter was following and capturing the IHME forecast, so we can actually look at them retrospectively, how they performed. So what we're looking at in black here, the dark black line underneath, those are the actual real data. And the blue lines are projected futures that did not come to pass from IHME. So you see this symmetric curve of things dying off. This is like COVID ending in June of 2020. And then you see some places where they had over predictions of the curve as well. And so I would, again, say that this is a pretty resounding example of a failure of forecasting. Interestingly, we should not even really be having this conversation right now, because Farr told us in his paper, the decline of the epidemic was less rapid than its rise, and the mortality was somewhat greater in the autumns of 38 and 39 than in the summers. So Farr is telling us the curves are not symmetric and telling us that there's this funny seasonal pattern that doesn't quite match the data. And then Farr tells us what Farr really did, which is by taking the mean of the deaths in the third and fourth period, the mean of the deaths in the fourth and fifth period, a regular series of numbers is produced. So of course, not surprisingly, if you do enough averaging, you can get something to look Gaussian, and Farr did that. Now, why did I spend a whole bunch of time talking about Farr's law? Because I think the failure of Farr's law tells us something interesting about the way we should be modeling and thinking about biological systems. And the reason is that we keep using models we know are wrong. And importantly, I'm not saying who either of these we's are here. I don't think none of the we's are in this room. They're in other rooms. But that actually might not matter, right? So Farr's argument is that maybe doesn't matter if we have the wrong model, as long as it produces the same statistical properties in our data. If what we care about is making a forecast or evaluating different kind of interventions, it may not matter. And there are plenty of you in the room who think, well, it absolutely has to matter. And that might also be true. So part of the reason I'm bringing up this issue of Farr's law is going all the way back to what most people would consider to be our first model of an epidemic. Not only in the paper does he talk about all kinds of things that we're still wrestling with, like seasonality and lack of symmetry in curves, but also this argument about whether we can take a statistical approach to modeling and focus on the data, or whether we actually have to understand the physics if we want to intervene and make predictions. But I actually think the problem is a little bit more complicated than this. And this is the latest version of the talk that I give, a version of this talk where I tell you that we shouldn't be using the wrong models. And I now have a version of the talk that says I'm either completely convinced that all the models are wrong and it doesn't matter, or that we do probably need to go after what the actual right models are. And so this is work that I've done with Laurent Dufresne and Jean-Gabriel Young, who are now at the University of Vermont, and Antoine Allard, who's at University of Laval in Quebec City. And it started with trying to explain this time series here. So these are weekly influenza-like illness cases in the United States from 1998 through 2014. And in red, I've highlighted all the parts of the curve where the early exponential rate is slower than the rate of exponential growth at the peak. So all the parts highlighted in red, the exponential rate as you approach the peak is higher than it is at the early part of the curve. Now, it turns out that there were no models on the shelf that will give you this kind of behavior. There were no published flu models that give you this kind of behavior where you have exponential spread and then faster spread, still exponential, but at a higher rate. So if you're familiar with seasonal forcing models, those models are almost always set up in such a way that you have subcritical transmission and then you have critical transmission. But once you get into that critical phase, the exponential rate will be fixed. And if you look at this, you might think to yourself, well, it looks like that happens every season. And it turns out that it does every single season, including all waves of the 2009 pandemic. And you might wonder, well, why on earth have we been using models that don't generate the data that we've observed? And this actually, we wondered this as well, and we submitted it to Science, and it got rejected. And it got rejected because they accused us of identifying our own paradox and solving it, but nobody else had pointed out the paradox before. And so it didn't count as that exciting of research. And we should have waited until somebody pointed out the fact that the data never matched any of the models that were out there until we came up with the solution for it. But what we did is to come up with a couple of different kinds of models that will generate this sort of behavior. The first kind of model that will generate this kind of behavior is if you actually have two different pathogen strains that are interacting with each other in a way that generates some additional nonlinearity. So you imagine you have pathogen A and B, and the probability of getting infected with B if you're infected with A is much higher. And so what might happen is A starts the spread, and it's spreading at its exponential A rate. And at some point in time, you'll have enough A infected individuals that B will realize a massive advantage, and you will see it kind of sweeping through the population. And so here what we're looking at, this is a paper that I was not a part of, where you have the force of infection beta, that's basically the transmission rate times the rate of mixing in a mass action model, versus the size of the remaining fully susceptible population for interacting contagions versus non-interacting contagions. And what you can see is that this is in the SIS model. In one of the models, you have a very sharp discontinuity into the state in which you have a non-zero fraction of infectious people, and that it happens at a different position. And it turns out there's a hysteresis loop underlying this that has to do with the buildup of the number of individuals that are infected with a certain number with one of the two strains. Importantly, this will give you this kind of speed up. So if there were two flu variants spreading that were interacting with each other, you could have early exponential spread and then later exponential spread as you go to the peak. There's another model that will do this, which is a model we were interested in after the Ebola outbreak, which is in a functioning society, if teachers get sick, nurses, healthcare workers, even football players, anybody that has to go to work will get replaced. Well, replacing people during an epidemic is a problem. Because if I'm replacing somebody who's sick with somebody that's healthy, I actually have more information about the local network state of both of those individuals that might give me pause about how I'm doing the replacement. Well, what do I mean by that? Well, if I go down sick, pretty good chance that I got sick from somebody in my social network. If I'm not sick, pretty good chance that nobody in my social network is sick, right? This is why message passing algorithms work very efficiently for setting epidemics on networks, because if I want to know if I'm sick, provably the best way I can do it is just ask all of you if you're sick, and then I can update my posterior probability of being sick. So what we're doing by replacing sick teachers is taking somebody who we know is healthy and probably in a comparatively safe part of the social network and rewiring them into a part of the network where we know there's contagion spreading because there are people going down sick that have to be replaced. And so what we're looking at here is the infection rate divided by the recovery rate, the proportion of infectious individuals on the y-axis, under 3 different models of replacement. So the lighter blue with the dash and the dots is the slowest replacement, and the darkest blue is the fastest replacement. So as we go from, vegetarian to meat, the replacement rate gets higher. 2 things to note from this. The first is replacing them can actually be good. So if you're replacing them faster than the transmission is happening, then you're slowing down the transmission. Once you slow down a little bit too much, or the infection is fast enough, basically what ends up happening is you have II edges getting rewired to be SI edges. And in the simplest version of this model that we're building, it has a network component. And so the rate of infection growing in the population is beta times the number of SI, or the portion of SI edges. And so you're effectively rewiring the fuel for the epidemic. However, once the rate gets high enough, you actually no longer have a smooth transition into, this is the SIS model, so the endemic phase. You have a discontinuous transition that actually looks a lot like this discontinuous transition. And you get the same kind of phenomenological behavior where you can have exponential spread and then speed up into the peak. There's yet a third model that can do this, and this comes from the social scientists, if you're familiar with the voter model. So the way that I decide if I'm going to get infected is I'm going to get infected with probability 0 unless at least 3 of my neighbors are infected, and then I get infected with probability 1. And we think this is one of the good models for how people decide to adopt different kinds of behaviors or retweet things or vote for different candidates, et cetera, is that it isn't really a smooth function. It might be a discontinuous function, but importantly, it's nonlinear in the number of your neighbors who hold a particular belief or who are infected. This is referred to as a complex contagion. And so here we have the force of infection divided by recovery for pathogen 1, and then for pathogen 2, as well as the stable fraction. And what I've done here is I've plotted both the interacting contagion version of this model, where we have beta 2 not equal to beta 1, and then the version of this model where you have no interacting pathogens, but you have this complex function that governs the probability of infection. And the reason that you can't see multiple lines is because they're exactly the same outcome. Turns out if you write all these equations down for replacing sick healthcare workers, for complex contagion, and for interacting pathogens, and you stare at them real hard, you can figure out that it's actually the same model. So what we showed in this paper that we published is that you can map all of the interacting contagion models and all of the sick healthcare worker replacement models onto a complex contagion model, and that they are all analytically equivalent to each other. And this is depending on, we wrote 2 versions of the paper. The first version I wrote is that it's all for naught and we should give up because we can't distinguish between these different kinds of models. Laurent and Jean-Gab wrote a version of the paper, which is what we ended up going with, which is actually, wait a minute, no, this is a great thing. We can provably show that with the wrong model, complex contagion model, I can model interacting pathogens. So you give me a time series that's generated with interacting pathogens, and you tell me it's interacting pathogens, I can fit my complex contagion model to it, and I will be just as good at predicting as if you had the correct model structure with respect to these interacting pathogens, and you use that one. And so maybe things actually aren't that bad, because while we might want to learn something as biologists about whether it's interacting pathogens or not, maybe from a public health perspective, if what we care about is demand on the hospitals, and so what we need a good forecast, we can use a model that we know is wrong, but that will give us forecasts that are reliable. And so it might be that there's actually some applied benefit of the issue associated with not being able to distinguish between the different kinds of models. I'm going to skip over this. There's yet a third example of this kind of behavior. And this is one of the few times that I know that I've been wrong about something, which is kind of exciting. I think one of the problems in the field that I work in is that nobody's ever really wrong about anything, right? Because it's like, I'm not claiming I have some proof of something. I'm claiming I have something that seems to be working just fine. And how working just fine it is up to me and the peer reviewers and the editors and the NIH. And so you're not really often wrong. And we were definitely wrong about this. So late January 2020, there was a paper in New England Journal of Medicine looking at the survivability of SARS-CoV-2 on different kinds of surfaces relative to SARS-1. And I stared at that and I thought I'd figured out the problem with SARS-CoV-2. The problem is that for all surfaces measured, there was no detectable difference in survivability except for cardboard, where SARS-CoV-2 survived for twice as long on cardboard as SARS-CoV-1 did. And I thought to myself, well, what have we done in the last 25 years since SARS-1? We have papered the world in cardboard over the last 25 years, and maybe that's what's going on. Maybe it's exactly the same kind of virus. It isn't really going to be a very big problem. We just have to make sure we put our mail in the microwave and do a bunch of other stuff like that because of the cardboard. And this turned out not to be the problem. However, a colleague of mine at Yale, Brandon Ogbunu, has been interested in these kinds of models for years, working on hepatitis and cholera. He refers to them as sit and wait. Wait stands for something, water, something, something, something. But the idea is that you have an infectious reservoir and individuals are visiting this reservoir and becoming infected. So you may have another infection process, but you also have this reservoir. So maybe it's a needle, maybe it's a contaminated body of water, maybe it's your Amazon Prime box that just showed up. And the model structure in the simplest version, this isn't quite the simplest version, but it's the simplest SARS-CoV-2 version, is that you have a susceptible class, an exposed class, asymptomatic and symptomatic class, recovered, and then you have this W box down here, which is the reservoir that's infected. And we got super excited because this model seemed to fit the data way better than the standard SEIR model. And we thought we were being creative about controlling for the extra degrees of freedom that are here, et cetera, et cetera. So when it turned out that we were wrong about the cardboard issue, we actually had a lot more of kind of an existential crisis than just being wrong about cardboard, because we also thought we were really right about this model being better. And I actually think we were right about the model being better. And you have to trust me that we did all the right sort of AIC type of stuff that we needed to do to control for the extra degrees of freedom. But I'm curious if anybody has any ideas why I might claim that we know this is the wrong model, but I actually think it still outperforms all the other models and for the right philosophical reason. Any ideas? I don't get to use my joke about asking questions very often because I'm still giving mostly Zoom lectures, but one of the things that I always tell people when I'm giving things on presentations is if you actually want people to answer a question, you have to out-awkward the audience. So what else might this W box be doing in the model? Yeah. Essentially, that's what it's doing. I have a fancier word for it, which is I think what this box is actually doing is capturing my uncertainty about the system, not coming from randomness, but coming from epistemic uncertainty with respect to the model that's generating the data. So we know that SARS is like basically an SEIR model with some asymptomatic transmission. Like that's probably what explains a big chunk of the variation in terms of what's happening. But we also know that it's definitely not just an SEIR model with some asymptomatic transmission. There's a bunch of other stuff that has to happen. And basically this box here is all the other stuff that's happening, especially because for different countries and different regions, we'll fit the parameters separately. So we actually kind of can tune the amount of other stuff that's happening in the model. And again, I think that this is an interesting aspect of this, because one of the things that this model will be very poor at doing is generalizing to other countries or to other disease settings. And I think that's an important consideration when we start to move into machine learning and considering the question that Farr was raising about whether we actually have to understand the physics or whether we can just learn things from data. Because a lot of what happens inside of this W compartment here is going to be associated with the kinds of things that where the generalizability would fail. But the generalizability would fail in kind of a spectacular way, right? Because it would be very hard to catch. And this is one of the things that I'm hoping to bring as a life scientist to the artificial intelligence field is what can we do if we know this kind of thing happens to design better experiments or design better ways of testing these AI systems to look out for these kinds of things. So an example that I did with the CDC is interested in doing genotype-phenotype mapping for influenza. And the simplest one that we could do would be oseltamivir resistance, which is a neuraminidase inhibitor that we still use quite regularly. And it's the simplest one because the CDC has boatloads of data where they've tested viruses for oseltamivir resistance coupled with genomes. And it's pretty simple. It's basically a single mutation that renders most any virus resistant to oseltamivir. We use neural nets to map genotype onto phenotype. And we come up with all kinds of creative ways of splitting the data out to try to convince ourselves that these networks are robust. And they pass all the tests with flying colors. Except for one test, which is if I train the data up through 2009 and test it against post-2009 data, it fails spectacularly. Now, why does this happen? Well, it's the same exact mutation that was causing the resistance in 2009, 10, 11 as it was in 2007 and 8. Turns out what happens, the way oseltamivir resistance evolves is that the lineages always, we don't exactly know why, there's some hypotheses about this, the lineages when they replace always start off as sensitive, and the resistant lineages spin off of the sensitive branch and then eventually get replaced by a new sensitive lineage. So the resistant lineages are always spinning off of sensitive branches which are replacing each other. Turns out what the neural net is doing is memorizing the phylogenetic tree, not learning what the cause of mutations are. It's memorizing the phylogenetic tree. And as long as you don't have a dramatic shift in the phylogenetic tree, it's going to do a great job. But if you have a new novel H1N1 replace your H3N2 lineage that's present, you're going to have a dramatic shift in how these sensitive and resistant lineages are distributed throughout the tree. And the neural net, which has doubled down on the phylogenetic structure, is going to fail. And we know this kind of thing happens all the time in biology, right? You have all sorts of interesting idiosyncratic patterns of how phenotypes map onto phylogenetic trees. One of the conversations that I was having with the math group this morning is how phylogenetic trees aren't even a very good representation of the way evolution happens. We know it's more complex than that. However, I think one of the things that we can bring in as life scientists to the AI question is our knowledge that this kind of thing will happen. And we can engineer systems that are either anticipating it, or we can design the kinds of data collection and experimental tests that specifically look for this. Second example of a PhD student, Juan He, and Juan is interested in predicting where SARS-CoV-2 genomes are sampled. Now, why is Juan interested in this? Part of the reason why is because it's a pretty easy problem to solve. There's a huge amount of structural information in the genome that is telling you basically where it was sampled. Just because you have a mutational process that's relatively slow compared to the movement of people. And so you have a buildup of mutations that are more like each other in the same geographic region. And in this case, we were more interested in things like problems with the encoder models for doing deep learning on genomes, problem with convolutional filters that are better for images than they are for genomes. So we wanted to pick like the easiest problem that we could think of. And this is one of the failings I've had as an advisor is I should have known that as soon as we think we've picked the easiest problem, we should pick something else because that problem is going to end up becoming a big mess. And that's what happened. Juan will have a much more interesting PhD as a result, even though it will take a little bit longer. And so what do I mean by this? Well, the model is amazing. 97% out of 7.5 million genomes, it gets the country right. And that's over all possible countries. So it's not like we're asking, did this come from England or everywhere else? It's which country did this come from? 97% correct. What about the ones that are incorrect? It turns out that the ones that are incorrect are almost certainly all mislabeled data. Now, what do I mean by that? I don't mean that somebody punched it in wrong. What I mean is that somebody got COVID in Paris, flew to London, they got swabbed in London, and the data is labeled London. And it's actually a Paris genome. So one of the things that we're able to show is that we can predict the errors based on the airline travel network. So if I want to know where the England genomes are most likely to be mislabeled, I can look at the airline network, and the probability of flying to different countries from England is a very good predictor of the genomes being, quote, mislabeled. This is related to some work we just finished in Nigeria, where we were doing a clinical trial around diagnosing pediatric malaria. We had an app that was built by physicians, takes the child's temperature, age, BMI, a variety of other things, and tries to assess whether there's malaria present or not. If you're a clinician and you work on febrile illness, you know that this is not a very good idea because malaria will probably present like dengue and chikungunya and influenza and RSV and everything else that's a febrile illness. Unless people are so sick that they don't need to be diagnosed by a clinician, they can just guess that they have malaria. However, they made a similar mistake, which is they wanted to have a gold standard of which to evaluate these clinical diagnoses. And so they did a PCR test, and for who knows what reason, also a rapid diagnostic test on all 5,000 children in the study. And the PCR and the rapid diagnostic tests only agree with each other 40% of the time. But this is where the life sciences enters, which is I actually think we can explain about 95+% of the disagreement. And most of you probably know what some of that is now, having gone through COVID. Most of the disagreement were PCR positive, RDT negative individuals that had unsuppressed HIV replication and low levels of parasitemia. So they weren't actually sick in the sense that they didn't have a fever. They were negative on the RDT test because they did not have acute malaria. But if you do a PCR or a blood smear, you'll find the parasite if you look hard enough. The rest of the difference is even more interesting, which is in about 10% of the cases, you've got the RDT being positive and the PCR being negative. That's not supposed to happen. And it's way more than what the false negative rate should be on the PCR test. And it turns out what's going on here is if you put the blood under a microscope, you'll see the parasites happily swimming around. And if you sequence the genome, the region that the PCR test is looking for is gone. And this is true in probably 15–20% of malaria cases now, I think, in Bangladesh. Why is it gone? It's gone because if you test PCR negative and they send you home, then you transmit malaria parasites to people. And so the selective advantage of those malaria parasites has gone up dramatically under PCR-induced test selection. It's part of the reason why at Northeastern, when I worked with the provost office on the design of our testing program, that we used 3 primer sets for different parts of the genome, because we were worried if we actually were successful at suppressing viral transmission at the university via high throughput testing, that we would place an incredible amount of selection pressure. And sure enough, we never published on this, but you see PCR primers start to fail, but not all 3 of them. So the point of this is connecting all the way back to Farr's law. I think there are plenty of interesting cases where some of the things that we know as subject matter experts can help us inform either the actual core development of artificial intelligence or machine learning systems, or the way in which we evaluate and test those systems. It's why the big initiative that I propose in the Institute is what we're calling Wet Lab in the Loop AI, which is to really seriously couple the life science lab researchers with the core AI researchers in a way that you have a feedback loop between both of them. I want to end by telling you a few things that I think we learned, not that we have the answers, but we learned we need the answers during the SARS-CoV-2 pandemic. And the first is about super spreading, but it's maybe not quite what you think about super spreading. So one of the things that's true about super spreading is that it can be problematic because you get lots of cases, and cases are bad. So classically thinking, super spreading is bad. However, super spreading can also be good. Now, what do I mean by that? Well, one, it can be good if you understand what's causing the super spreading, because, for example, if it's unsafe burials in the case of Ebola, if you have safe, respectful burial programs, you can rapidly bring an Ebola outbreak under control with that measure alone, because you're reducing the super spreading events. And the super spreading is what's propping the disease up above the critical threshold for an epidemic. Even if you don't know where the super spreading is coming from, that's still a good thing. And that's a good thing because if you have a disease like influenza, where you have relatively little super spreading, then there's almost nothing you can do aside from locking down and waiting until there's a vaccine, because flu is going to march along deterministically with R0 equal to 3 until it runs into a vaccine or a lockdown. Flu, the distribution of secondary infections, is basically Poisson with lambda equal to R0. So there's no super spreading. Doesn't mean you never have a large cluster of cases, but it means that the super spreading does not contribute meaningfully to the dynamics of the system. If instead you're faced with the Wuhan strain of SARS-CoV-2, which has a pretty similar R0 to flu, but instead it has that R0 because you have people giving it to 30, 40, 50, 60, 100 other people, and a lot of zeros attached onto that. One, it means that if you can identify the source of super spreading, you can have a large effect on the system. But the second is, and this is one of the things that has been very interesting, my background's actually in population genetics, and so one of my favorite pastimes is to find the stuff the PopGen people knew 30 years ago that we have missed in Epi up until then. And one of the things that some of you didn't miss, but I missed, is if we flip the problem on its head and I say, what is the extinction probability of this spreading process given a certain mean and variance in terms of the number of transmission events, the extinction probability goes up with the variance quite rapidly. And so if I have a lot of super spreading and a relatively intermediate R0, any individual chain of transmission is more likely than not to die out on its own with no intervention. And it also helps us learn something very interesting about the introduction processes. Namely, if you remember, there was this case in Seattle, and Trevor Bedford's group put out this tweet saying that there'd been 6 weeks of community transmission. That's based on a Poisson model of how transmission works. If you include the super spreading, you're actually very unlikely to see 6 weeks of uncaught transmission because you have 1, 2, 3 cases. Instead, what's going to happen is it's going to go extinct because you'll have 1 and then 2 and then a bunch of zeros and then no more transmission. There's a huge amount of evidence in the genomic data up and down the West Coast that this happened. Multiple introductions of SARS-CoV-2 that went locally extinct. Instead, if you see a super spreading event, it's actually much more likely that the origin or the origination of this is only 1 or 2 transmission layers deep, especially if you hadn't seen cases before. And so what is much more likely, and this turned out to be the case after we got the genome data I'm saying we, I wasn't a part of this. We, like the capital we, like all the scientists, that the introduction in Seattle was really just 1 or 2 transmission events and then the super spreading. So super spreading gives us a whole bunch of things that we don't get without it. However, there's a bit of a problem. And this is the first thing that I think we learned during SARS-CoV-2. You probably saw people fighting about herd immunity thresholds on the internet during COVID. Now, my personal opinion is that the herd immunity threshold does not exist in the real world. It only exists in infinitely large mathematical models that don't exist in the real world. And so we shouldn't really even be talking about the herd immunity threshold. That's my first opinion about herd immunity thresholds. My second opinion about, it doesn't mean we shouldn't study them as mathematicians. I think it's very interesting and rich and a whole lot of fun, and it does tell us something about the biology. We just shouldn't try to operationalize it. Maybe be a little more clear about that. However, even the herd immunity threshold often has pretty simplistic assumptions about the way super spreading works. So it turns out, like, if we look at something that has a mean and a variance at first and second moment that are different from each other, so we relax the Poisson assumption, we can say something about what will happen to these chains, about what will happen to the herd immunity threshold. However, if you have third, fourth, fifth moments in the distribution, they affect the probability of establishment with alternating signs and in a very interesting and complicated way. And so if you think that your distribution of secondary infections doesn't just have a meaningfully first and second moment, but also higher moments, then you, as far as I know, nobody has actually ever come up with a closed form solution of the herd immunity threshold under those models. However, it actually gets worse than that. It's worse than that if you believe it. And actually, this is a bigger problem for everything, for how we would think about epidemic risk, how we do our modeling, et cetera, et cetera. All of this stuff that I've been telling you about, including the super spreading about the transmission chains and et cetera, all assumes one very big thing that I don't think we know is true. And in fact, one of the conversations that I was having at dinner and then this morning is that we probably can reason our way into thinking that this actually shouldn't be true. It assumes that the in degree and the out degree in the network distributions are symmetric. Assumes that if I have a high risk of getting infected, I have a high risk of spreading the infection. Now that may be true for some people, right? So for teachers, that might be true because you're up in the front of the room projecting. If it's a respiratory virus, then you're having lots of one-on-one interactions with your students in close quarters. So you might actually be at high risk of getting infected and at high risk of transmitting. But maybe if I work in the custodial team at the hospital, I might have a very high risk of getting infected, but I might have a low risk of transmission, although you could make up the opposite as being true. The question is, does it matter if we decouple incoming risk from outgoing spread? And this is work that we did with Chris Moore and the same cast of characters that I mentioned earlier. So we thought about 3 different kinds of scenarios. The scenario that we mostly all consider, which is that if you have a high risk in, you have a high risk out. If you have a low risk in, you have a low risk out. Another scenario where you have a high risk in, but a low risk out. And then where you have the low risk in and the high risk out. But does this matter? Absolutely, and in kind of a spectacular way. If your in degree distribution is Poisson, it does not matter what the out degree distribution is, even if it's power law with no second moment defined such that there shouldn't be an epidemic threshold. The system behaves as though it's Poisson. So almost all the dynamics of the system are governed by the in degree, especially if that in degree is Poisson. How many people could point me to a piece of data looking at in versus out degree in human social networks? I could probably dig one up from a telco company if I really tried hard and showed up with a bag of money for the compute charges, but they're not out there. If you think about the way our social networks work, how often we change locations during the day, we change different people that we're around, et cetera, it probably shouldn't be the case that our baseline assumption is that the in degree and out degree are the same. But there's a very good reason to believe that it will matter if they aren't. Especially because what we often see are the out degrees, right? So if you think about contact tracing studies and we look at the number of secondary infections, that's really heavily driven by the out degree. And what I'm telling you is that that doesn't matter if the in degree is wildly different from the out degree. I think there's probably, if somebody in the room that's smarter than I am, I know that most of you are, sat down and thought about it, you could probably look at the right kind of contact tracing data and back out whether the in degree and out degree were symmetric enough that you didn't have to worry about it. I bet you could come up with a way to solve that problem. In fact, that may already be a solved problem with branching process models with different kinds of variants. And again, I haven't even mentioned some of the other problems with this, which is, again, all of these models assume, this is my qualifying exam question that I'll ask the network science students next week, if any of you are listening to this on Zoom, which is, what kinds of things break our intervention models for networks that we know occur in the real world? And one of these is that you shouldn't have strong degree-degree correlations in terms of who's connected. And we know that happens. We know that people with lots of social contacts often gravitate together in dense sets of the core. That's why we work on these core periphery models. We basically don't know any, we know a little bit analytically about what happens in there, but we certainly don't have anything that looks like the herd immunity threshold. And so even if we ignore the fact that the virus evolves, ignore the fact that behavior changes, ignore the vaccine, ignore all the stuff that we know is going to happen, and we just do what the mathematicians who said that the herd immunity threshold was going to be at 40% tell us to do, it still doesn't work. It also doesn't work for another very interesting reason, which is, under this branching process model where we look at the multiple moments of the distribution and we can look at the extinction probability, we can also get an estimate of the final size. And our estimate of the final size for COVID is 40%. But if we actually run the simulations, you don't get 40% because you have, if you run 10,000 simulations, you infect 40% of the people most of the time. Instead, what happens is what you'd expect to happen in a nonlinear dynamical system, which is 40% of the time everybody gets sick and 60% of the time nobody gets sick. And so the only realized outcomes in any one particular population or run is that everybody gets sick or nobody gets sick. And if you average over lots and lots and lots of them, then you get to this final size of 40%. And this is, of course, also what happened with SARS-CoV-2, right? Is that you have lots and lots and lots of introductions essentially raining down all over the world. And then we removed a lot of the mobility. So you had lots of closed populations with lots of introductions raining down, raining down, raining down. And we were used to thinking about these kinds of models in the wrong sort of context with respect to that system. In fact, I distinctly remember, I'm not going to blame it on anybody because I don't remember who said it. I was just getting ready to blame it on the CDC, but I don't know if they even said this. Somebody said we can stop tracking transmission once local transmission is established, especially if they're imported cases, because imported cases won't matter once you have local transmission. And I actually think that's the biggest thing that's been debunked during the pandemic, is that most of what happens are metapopulation dynamics, and the reimportation of cases is basically all that's driving most of the dynamics. That you have something like the Alpha variant get a hold in London and lots of exported cases into the surrounding areas. It's what happened in Wuhan, that you had it get established in one of the world's mega cities and then infecting lots of surrounding areas. It's what happened in northern Italy. And we actually think that some of the idiosyncrasies in terms of how the metapopulations are structured are what led to the differences in the global outbreak dynamics. For example, in Italy, in this northern part of Italy, you have a lot of big car manufacturing plants, and you have a lot of people that live in the same town and go to work in the same place. That's a very different situation than in Wuhan, where you have more of a Boston-like situation with these sort of rings of suburbs out around. And it might not necessarily be that common for you to have your neighbors working in the same building as you. And so it turns out those differences in the way the social networks are structured, when you layer on and assume that the metapopulation dynamics are what's going to be driving things, ends up really mattering in terms of the kinds of dynamics that you see and what happens when you turn off, for example, the mobility and how quickly you might expect things to locally go extinct. And we have some data to show you this. One of my favorite examples is an estimate that for the Alpha variant in Scotland, it was 300 separate introductions that ultimately led to the epidemic curve. That's the other thing that's problematic with herd immunity threshold and R naught and just about everything else is you assume that you have a single infected individual dropped into a fully susceptible population. We normally say that the susceptible population part is a problem and we can kind of ignore it, but we don't spend enough time thinking about the fact that it's actually not the case that you have a single infected individual. It's that you have basically n infected individuals raining down proportional to the connectivity of the cities to whatever the source of infection is. And that's a very different set of initial conditions than is assumed under most of the models. The last thing that I want to mention is I think the problem is even harder than this, which is even if we pay attention to the metapopulation dynamics, most of the within metapopulation dynamics we still think of as, okay, we'll just use the SEIR model. We might not even use network structure because we think about, okay, if I tell you that the import-export dynamics and the regional mobility, that's what's governing it, we'll just ignore the stuff that happens inside of the nodes, right? So I'll have every census tract, and I've done this, I've published models on this, so I can say it's wrong. Every census tract is the node. We only model mobility between the nodes, and inside the node, we just do a SEIR mass action model because it's fast, and it probably doesn't matter. Turns out we think it matters immensely. So this is the cartoon of what happens when you drop a single infectious individual into a well-mixed population. You actually get a curve that doesn't look that different from what Farr suggested, right? It kind of should go up and come back down and be pretty symmetric, especially if you look at the average over lots and lots of different runs. However, we know even within a node, even within a census tract, that people interact in a hierarchical community structure. They share workplaces, they share schools, they share playgrounds, they share different households, or they don't. And so you drop a single infected individual into this kind of population, and probably it will spread, but it's not exactly clear what that curve is going to look like. Now, why do I care what the curve looks like, aside from promising you that this was a talk about the shape of epidemics? Well, I remember very strongly in April 2020 being all over the sports radio in Boston telling people we needed to flatten the curve by the lockdowns to protect the hospitals. Well, how do we flatten a curve if we don't know what the curve looks like? Like this curve, I can flatten that curve by a lockdown. Can I flatten whatever goofy looking thing is going to come out of this model with the lockdown? Hint, the answer is no. So there's actually some interesting theory that Vittorio Colizza and Alessandro Vespignani worked on, and an interesting paper by Duncan Watts et al., who looked at this in terms of resurgent epidemics in a hierarchical metapopulation. And what they showed is that they have a way of adding layers. So imagine you have, if you're familiar with like a stochastic block model, you might have different hyperparameters that govern your membership in different communities, and you can add more non-zero parameters. So this is where you just have one layer. This is what the outbreak looks like, just that Farr curve. That curve we can flatten. This is two layers. Now these are stochastic simulations, and you can squint and maybe it looks kind of like there's two little waves there. And if you have three layers, you can squint and it kind of looks like there's a few there. And it turns out that as you add more layers, you get more and more waves. And if you have noise, you go from having waves to just a big smear. And interestingly enough, this is, if you haven't read this paper, this I think is the coolest paper that's ever been written. It's by Lisa Sattenspiel and Deanne Herring, anthropology professors. Also pretty hardcore computer scientists with respect to early agent-based models. They're still doing a lot of very cool stuff, but they're very early in the agent-based modeling space. They took data from the Hudson Burr Fur Trading Company during the H1N1 1918 pandemic. And they used people not showing up in the logbooks as a way of trying to get an estimate of who was getting sick or not. They used this to parameterize an agent-based model. And they showed that the different kinds of community structure that was present in different parts of Canada correspond to the number of waves that they got in the fur trading camps. So you get this kind of dynamics associated with the hierarchical structure of the way people were moving around these fur trading camps. So what we did is we took data, this was early in 2020, from Italy and China. We measured some proxies, they're very poor proxies, I'll admit, for the amount of clustering present in different cities. And then we measured the epidemic intensity. So you have to come up with some way of deciding the difference between these curves. And the way that we did this, there's a measure that has been proposed before, which is just to think about this as the possible states the system could take on. The height of the bars represents the state that it's in, and the possible states are along the x-axis here. And so I could just do something very dangerous, like calculate the Shannon entropy of this and call that the 1 minus that the epidemic intensity. It's in the literature. We can talk about whether there's a better way to do it. What we showed is that across China and Italy, over multiple orders of magnitude in city size and epidemic size, there's a strong relationship between the amount of social crowding, which is our proxy for the degree of hierarchy, and this epidemic intensity. So essentially what happens is in a city like Sao Paulo, which is really a giant mega collection of lots of neighborhoods, you will never have the epidemic go extinct because it's just going to jump from subpart of the graph to subpart of the graph to subpart of the graph to subpart of the graph forever. And it'll be a big, long smear of cases. Whereas in a city like Manaus, which is quite large, but very isolated and rural, you have a much more connected set and you get a curve that sweeps through like this. Now, does this matter? Turns out that it does matter. So now what we're going to do is we're going to simulate outbreaks on either the hierarchical or the standard Farr's law type of curve. And then we're going to impose some interventions and see what happens. So on the left here, this is, on the left, this is the sparse network. This is basically a single layer in the community. These are a whole bunch of outbreaks. The gray is the sort of median over a whole bunch of different outbreaks. On the right, this is where we have more layers in the metapopulation. And you see that you get some that only get one layer and you get a little curve like that. And you have some that are clearly getting multiple layers and you're getting this sort of smear of cases. And now what we're going to do is we're going to lock down people in their households. So what happens? Well, if we lock down people in their households in the single layer community, the curve does what it's supposed to do under the flatten the curve model, which is to go down and shrink out. If we do it in the metapopulation case, it actually turns out it does shrink in, but it also gets higher. And now why does that happen? Well, we know that a little bit of this is baked in, but we know that the household transmission rates are higher. When you're not locked down, the proportion of time you spend outside of the household is much higher than when you're locked down. And so what we do is we concentrate all of these social connections into a very short period of time, and you get a rapid burst of infection that will quickly burn out, but with a much higher peak incidence than in the other model. And so if it's the case that we actually care about the hospitals getting overwhelmed, it isn't necessarily true that lockdowns will flatten the curve and push out the peak to a lower later level. It could actually intensify the curve and cause you to have an earlier, higher peak. The area under the curve will still be shorter. You will still save lives under this model, assuming that the hospitals don't get completely overwhelmed. The last thing that I want to say is something nice about IHME, although it doesn't sound like it from this tweet. So it says, why am I so critical of IHME? Because this is a group that since day one of the epidemic has prioritized publicity over accuracy. It's unfortunate because they've done some great public health work in the past. And I actually think that this is something that should be much more critical about the work that's being done outside of IHME. I think it's our fault, and I'm putting myself firmly in the we category here, that IHME even had relevance in this case. So what did IHME do? They did what weather forecasters do, which is they made hyper-local forecasts that people cared about. Like, I don't care about how many COVID cases are in Portland, Oregon right now, unless maybe I have somebody that lives there. I'm getting ready to travel there. I want to know how many COVID cases are here, because that's what is going to affect my behavior. It's the same with the weather forecasts. They also told us when it's going to stop. They're wrong about it, wildly so. That's the other thing with weather forecasts, is that we don't say carry an umbrella and I will tell you at some point in the future when to stop carrying it, but I'm not going to tell you when that is. I'll just call you one morning and tell you it's okay to stop. What they built was the kind of user experience and user interface that people would actually use to make decisions. And the CDC actually published a paper on this just a couple of weeks ago, showing in places where people had better information about local infection rates, they did a better job of keeping themselves safe, right? Like, people don't want to get sick. If you give them the resources to not get sick, and those resources can't be do this forever, never send your kids to school, nuke your mail in the microwave, et cetera. Like, those aren't options that people are going to take for very long. If you give them the resources, the masking, the testing, et cetera, as well as the local information they need to make decisions, people will by and large do it. And so I think the thing that IHME taught us, and that I still don't see enough of, is if we can provide the hyper-local information, people will make decisions to keep themselves safe. They also have to have the resources and the sick leave and a variety of other things that not everybody's going to have. Now, why do I think that that matters? Well, a lot of what we're trying to work on here is justifying why we need better models, better data, better machine learning methods to drive the accuracy in space and time increasingly granular, to have philosophical arguments about whether we need to understand the underlying physics versus just the statistical nature, to talk about the things that Farr is talking about. All of the things that we are doing, those bills can be paid in terms of the benefits that will come for individuals if we deliver to them actionable information at the local level that they use to keep themselves safe, to keep the schools open, to keep the businesses running. The flights won't be on time, but other things will be running just well enough. So I think that is, to me, one of the things that we need to take home with us. So big lessons in my mind is that everything's about metapopulation dynamics. We need more empirical data on real world networks. Super spreading is massively underappreciated. I think it's also understudied on the mathematical side of things. Some of you are going to disagree with me, but by and large, I think it's pretty understudied. Behavior. And then the number one thing, and this is the IHME thing, and I credit this to Professor Peter Dodds, who always says, never bring data to a story fight. And that's the thing that IHME taught us with respect to how you generate the kinds of tools that people will use. It doesn't actually matter if they're any good. It just matters if people think they're useful. Now, what we can do as people who understand how to build tools that are good is to copy this approach in terms of justifying why it is we need increasingly fine spatiotemporal resolution, why we need to invest in wastewater surveillance, high throughput genome sequencing, artificial intelligence that can do data disaggregation. All the kinds of stuff that we're working on can drive us towards the sort of endpoints where we have an IHME style product that people are addicted to and use to make masking decisions, and that saves lives and keeps schools open, but does so in such a way that it's actually accurate. Because one of the things that ended up happening with IHME, and it's what happens when you have inaccurate forecasts, is that people will stop paying attention to them, right? So the clock starts ticking, people will figure out that these forecasts are inaccurate and stop paying attention to them. So with that, I want to thank you all again for inviting me. I've very much enjoyed the conversations this morning, and I'm excited for the talks this afternoon. And if you have opinions, good, bad, or ugly, about artificial intelligence and life sciences, I would love to hear them via email. And thank you very much.
Okay, so let's open the floor. Get folks here. Go ahead, either come to the mic, or I think that's the best way. But go ahead. Go.
So that was great. So there's this little short on recorded lectures from Feynman. There's it's a 5-minute thing on understanding and prediction. And he uses, he says you go to Central America and they have this little device that tells you where all the planets are going to be and stuff. But the problem is it doesn't refer to anything about planetary movement or planets or anything else. It just predicts where the lights in the sky are going to be, but it's very predictive. And so the first guy that comes up and says, well, I have all these little things and they're flying around, but he can't predict where they are, they throw him out, right? They take him to the top of the pyramid. So there's I think there's a lot of that going on. I don't think that the explanation and the prediction necessarily line up right away from the point of view of underlying understanding, because with improved underlying understanding does not follow improved prediction right away necessarily, right? And there's a bunch. So once you're in the pure model piece, then you have there's another story about there's this paranoid guy and he says, well, people are always following me. And you say, well, why do you think that? He said, well, every time I turn around, there they are, right? So then you say, well, that's just regular people. And they come and they just start adding things onto their explanation, which explains why they're there and why they're not the same people all the time. But it makes sense to this guy, but it's an unbounded model. And it reminds me of that W box and this other thing is it gets so locked onto the first model and that you can't possibly change that when you add the W, you don't notice that you just explained why everybody's following you all the time. It's the first model that might be wrong. So what I'm coming to is the in degree versus out degree is a problem, right? Just as an example, a more concrete problem. And if you simply add, for example, if you add the idea that when stuff comes in on the in degree, it takes a while for anything to happen before any particular out degree happens. And there's a different out degree at the time that the in degree you're counting, is not the same thing, right? So you have an instantaneous graph. So it could be that the graph itself as an abstraction of this contact is not explanatory. It is not the planets, it's the little widgets inside the things, right? And so without adding, so mobility keeps mattering, behavior keeps mattering. We don't know why. That's adding the planets in, right? But it reduces necessary, perhaps, the thing. So my question to you is, how do you propose with your AI to avoid the paranoid model elaboration problem, to add and test the things that might be important, even when they wreck your prediction for a while, and move toward a more full understanding so that we can both get prediction and an understanding going forward? Like, what is the balance between the intelligence and the representation that you attach it to, for example, like the graph seems obvious, right? Except a simple thing like a time delay could completely make it wrong, right? So if you're going to set this program up, how do you go about this improving both prediction and understanding over time? How do you propose to think about that?
Yeah, great question. Which I would sort of say, maybe to stay back, like, why am I doing any of this stuff, and does it matter? There's a longer version of the talk, which is probably hard to imagine given that I went a bit over time. But one of the earlier studies I did was to take an information theoretic approach to looking at infectious disease time series. And the reason I did this was that we were all at the time pretty bad at forecasting these things. And the hypothesis was that we either didn't understand the physics or we didn't have the right data. And there's, of course, a third possibility, which is there are some time series which we're just not going to be able to predict any better than sampling from the historic data in the time series. No model, no that stuff's going to matter. And that turned out to be the case for infectious diseases, is that it seemed to be something about the information theoretic properties of the time series themselves, more so than the underlying generative models or the data and the observation model problems. However, this led me then to think about, like, in the case of this model with the reservoir, this is a simple case where I know it's a problem. There are a lot more complicated models that I personally have used that are probably also problems, but I'm not smart enough to show you that it's a problem. So what I'm sort of thinking about this is like the canary in the coal mine, so to speak, that we can come up with some pretty simple scenarios in which we provably won't be able to tell the models apart, and or where we could have a model that really seems to be working quite well, and we're really convinced of the mechanism because I labeled the W box infected needle, when in fact it could be one of N other things that would give us the same kinds of dynamics. And I actually think that that question is at the core of artificial intelligence, because I think we have an assumption that we will either be able to learn how the system works from a physics perspective by training AI on data, or we'll be able to extract the governing equations from the data using AI. And I don't think that either one of those are going to be true. And I think if we can extract the governing equations, then I think there will be many other equations that will generate equivalent dynamics. So we will have to do something else. And this is what I'm hoping we'll be able to make progress with on the experiential side of things with this wet lab in the loop process. Is I do think we can make different predictions under different, we have 10 different models that I claim will generate the same time series data. They don't generate all the same predictions under certain experimental conditions. And I think that's where we're going to be able to make the progress. But I know I agree with you. I mean, the title of this talk could be basically like the existential crisis I've been having for the last 10 years about what it is that we're doing here. If we can't actually pull out the models and we can prove that the data won't actually tell us enough to make future predictions in many cases.
Thanks for the great talk. I was curious if you've thought about, if you've thought about reservoirs at an individual level and how that might impact some of the community structure patterns that you've seen in some of your studies. So for either immunocompromised individuals or at the risk of raising a conspiracy theory, more ubiquitous long-term infection that like via long COVID and those types of things with emergence. And there was a nice paper recently and pointing out like the ladder-like surveillance and the deep branch surveillance that sort of and that might happen at different scales, right? You know, Omicron is a big deep branch, but there might be medium deep branches or something like that.
Yeah, no, so I think maybe the question, if I say it back, is a little bit about reservoirs and what that might mean for something like Omicron, but also maybe what that means in terms of transmission risk. One of the things that's a big challenge is with super spreading events. Everything that I've told you about so far with super spreading events assumes that the super spreading is about the social network. And we know that there's a lot of super spreading that's about intrinsic properties of individuals in terms of their immune system and how quickly they clear the infection and their ability to have asymptomatic infections that are long-lived. And so, again, as much as we don't know and understand about super spreading as it pertains to the social network, we don't even necessarily know a priori that super spreading will be caused by the social network. And so if the super spreading is happening because of properties associated with the individual, assuming that those aren't correlated with properties in the social network, then it's a much different situation, at least with respect to the interventions. It may not be a different situation in terms of, well, it probably would be in terms of the probability that it dies out or not. I think the Omicron piece is a really interesting one in terms of what happened there. I'm still waiting for this adaptive dynamics paper for COVID variants that somebody I'm hoping is going to publish. I don't have anybody in mind. I just keep waiting for somebody to publish this paper. We're again talking about this afternoon, where if you ever wanted to study something where you had an evolutionary process with a temperature that was being raised and lowered, like we've been raising and lowering the migration temperature on the planet in a well-documented way for the last 3 years, and we have 14 million genomes that have been sequenced, like there's something that we still don't understand about what happened with Omicron, because it could have just been really bad luck, but the wastewater data that I know the best, like there are lots and lots of variants that show up in the wastewater that never show up in any of the human genome sequences. We think they come from humans because they sequence the other genetic material, so it's not like this is an infected rat or deer or something that happens, but These are people infected with variants that don't show up in any of our data sets, that are wildly different from any of the things we've seen, and they just don't seem to be causing problems. And so whether Omicron was just a jackpot in terms of you get enough people infected and evolution is going to find something that maybe we don't want it to find, or whether there's something else that we missed about how the evolutionary patterns were happening, I still feel like that's a pretty big open question.
Thanks.